Hey everybody, it's group C33 here, and our research topic was how the FANCM gene has been linked to breast cancer. Breast cancer is the second most lethal known type of cancer, with over 3 million reported cases in the US annually. Studies into the inheritable cause of breast cancer have diagnosed loss-of-function mutations in genes involving DNA repair. Exomes are nucleotide sequences encoded by a gene that remains present within the final mature RNA after introns are removed by splicing. Exomes represent only approximately 1 to 2% of the genome, but contain approximately 85% of known disease-causing genes. Scientists have begun looking into the Fanconi anemia complementation gene M with the nonsense mutation C.5101C greater than T. This mutation has been found to be more frequent in patients with breast cancer.
Exome sequencing of breast cancer families in the genetically homogeneous Finnish population was used to identify recurrent alleles associated with cancer risk in previously unidentified breast and ovarian cancer susceptibility genes. The study was divided into 3 different phases or groups, with a total of 3,166 breast cancer cases and 2,090 healthy control participants. Phase 1 was made up of breast cancer type 1 and 2 negative familial breast cancer patients. Phase 2 consisted of controls, and Phase 3 consisted of breast cancer patients, additional familial cancer patients, and healthy population controls. After combining results from the 3 phases of genotyping of breast cancer cases and controls, a significant association with breast cancer was found for the FANCM nonsense mutation. Approximately 2-fold increased mutation frequency was found among breast cancer cases, 2.9% compared with controls, 1.4%.
In this figure, all of the OR ratio are above 1, which means the positive correlation with the breast cancer and the nonsense mutation. Most of the p-value are really small, which is statistically significant. This is a pedigree. All of the people have a mutation. Half of them have a cancer, half of them have not. Basically, having the mutation increases the probability of the cancer.
And to link it back to class, there are 2 types of traits, sex-linked and autosomal traits, and this has more to do with sex-linked traits. And dominant and recessive genes, and this has more to do with recessive genes. The most relevant information is found in the chromosomal theory of inheritance in lecture 4.3. We worked with Punnett squares, and we crossed P's using Punnett squares to understand recessiveness and dominance and inheritance. We also worked with Drosophila, or went over Drosophila, a type of fly, and studied its body color and wing type, and how those were affected by inheritance. Homologous recessive is more common with a more isolated population, like the example given of the breast cancer patients. The FANCM is more common by the same theory, and that is shown by this experiment. When a homologous recessive gene is paired with more of its own type, it becomes more frequent than in other instances. And shown by these 2 test crosses, there are 2 heterozygous genotypes crossed in the first picture, and in the second figure there's a heterozygous and a homologous recessive. And in the homologous recessive, there are more homologous recessive genotype inheritance.
To sum up, a large amount of data supports the idea that patients who suffer from the FANCM gene mutation have an increased susceptibility of gestating breast cancer within the familial population. The experiment comparing the familial population to the control population shows that the gene is recessive. The recessive nature of the FANCM mutation shows that the gene mutation will show up in more isolated populations, such as in Finland. Further research is still needed in order to identify all agents that lead to breast cancer, but the FANCM gene and other gene complexes like FANCM are the starting point to discover the original cause of cancer.